DNA and RNA, the blueprint of life. Just a little touch of humor. In 1953, the structure of the DNA molecule was first described. So the goal of DNA and RNA, or the purpose. DNA and RNA exist to act as the code for all the proteins that are made in your body. The chemical reactions that we've been talking about, like respiration and photosynthesis, run according to these proteins. We've looked at proteins in the following reactions. Like I said, photosynthesis and respiration. Also in mitosis and meiosis. Proteins also determine the physical traits that you have, like your hair color, your eye color, the shape of your nose, whether you have a dimple in your chin or not, and how your body reacts to your environment, like whether you have allergies to certain things. Basically, proteins do everything, and DNA is the code for how to build those proteins. DNA stands for deoxyribonucleic acid. The next couple questions on your list ask you in this slide to tell me what you think chromosomes are and genes are, and where the DNA is stored in the cell. If you take a look, this part down here represents a strand of DNA. One single strand of DNA gets twisted together to form the chromosomes that we talked about in mitosis and meiosis. These chromosomes are stored in the nucleus of a eukaryotic cell, which is what you and I and all mammals and plants have. A gene is just one segment, one piece or one portion of the DNA strand. That particular piece codes for one particular product, or is the code for one particular protein. Some facts about DNA. It was established by these two guys here, James Watson and Francis Crick. They were the ones, along with other scientists who are not usually acknowledged for being part of the process, but they were the ones that came up with this is what the structure of DNA looks like, and is what we see today as that twisted ladder that looks like this. That twisted ladder shape is called a double helix. Double because it has two sides, and helix because it twists. To review, DNA is the code for your genes or your traits. DNA is made of repeating subunits, repeating pieces, called nucleotides. A nucleotide consists of these three parts. This part, and this part. And it's connected on the other side of the ladder to another nucleotide. The connection between the two nucleotides is made by hydrogen bonds. So this is the same DNA molecule, but in its twisted ladder or in its double helix shape. The blue and whatever that color is, part of the DNA, that acts as the outside pieces of the ladder, and then these two pieces act as the rungs. So what is a nucleotide? A nucleotide is made of three things: the phosphate, the sugar, pentose meaning it has 5 carbons, and the nitrogenous base. The nitrogenous bases are nicknamed A, T, G, and C. So the rungs of the ladder are formed by the bases. One base is connected on the other side of the ladder to another base, again by hydrogen bonds. The sides of the ladder are formed by the sugar and the phosphate groups. There are some rules about how the two sides of the ladder connect to each other. The chemical adenine always bonds with thymine. The chemical guanine always bonds with cytosine. In this way, they form kind of a puzzle. So you can see that puzzle is represented here with the corresponding shapes. If you have a pointy part here, you have a place for that pointy part to go here. So this is thymine and this is adenine. If you have a round part, you have a place for that round part to go over here. Cytosine, guanine. And again, these dots in the middle represent the hydrogen bonds that actually connect the two nitrogenous bases. One more time, on the outside of the ladder, you have the deoxyribose, that's the sugar part, and the phosphate group. So, following these rules, you can decide, or you can come up with, if the one side of DNA has the following nitrogenous bases, you should be able to come up with what the other side would say. So for example, if one side of the ladder went in this order, A, T, A, T, C, A, T, G, C, and G, 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 you should be able to figure out what the other side of the ladder would be. I'll get you started. A always bonds with T. T always bonds with A with T, T, A. C always bonds with G, A with T, T with A, G with C, C with G, and then these would be 3 C's. In order to remember this rule, which it will be important for you to remember, I use a little mnemonic device. Mine is at my house. The car goes in the garage. So at A bonds with T, and car goes with garage, C and G. One of the questions on your list of questions for this is to come up with your own mnemonic device to remember this base pair rule. So again, here's the answer to what we just went over about the base pair. A with T, T with A, C with G, and G with C. In DNA, that's the rule all the time. Just to review, this whole part here is a nucleotide. It consists of 3 parts, the phosphate and the sugar, which form the outside part of the ladder, and then the nitrogenous base, which can be either A, T, C, or G, and that is bonded with hydrogen bonds to the other side of the ladder, and 2 bases together form a base pair. One of the questions on your set of questions asks you to watch this video. Read that question now, and then I'll start the animation.
DNA contains 2 strands of building blocks called nucleotides, arranged like a spiral staircase. Each nucleotide includes 3 parts, a phosphate group, a sugar molecule, and one of 4 bases, adenine, guanine, cytosine, or thymine. The sugar-phosphate bonds form the double backbone of the molecule, the handrails of the staircase. But we find the genetic key to DNA in the steps of this stairway, the nitrogen-containing bases. These bases link up using hydrogen bonds in a very specific way. Adenine will bond only with thymine, A to T. Cytosine only bonds with guanine, C to G. While these basic pairings never change, the order of the pairs along each strand varies greatly from one species to the next. In this elegant design, we see how nature stores the instructions to build all living things.
So again, the question was, what is the genetic key? And if you were listening to the animation, you heard the genetic key was the 4 bases and the order that they come in. So here's how the code works. The combination of the A's, T's, C's, and G's determine the traits you have. For example, CAT might yield or might give you purple hair, whereas TAC might give yellow hair. Think of the bases of DNA like letters. We use letters in our alphabet to form words. The words are like the genes. The genes, or the words, form sentences. And from those sentences, we have endless combinations or endless possibilities of what any organism, what traits any organism can have. RNA is similar to DNA, but not exactly the same. It doesn't quite have the same purpose. RNA is the messenger. It carries the message, or the code that the DNA has, to the ribosomes. Remember when we were studying cell structure? Ribosomes are located out in the cytoplasm, sometimes along the endoplasmic reticulum. And these ribosomes are where proteins are actually made. And then those proteins are, what makes your body function the way it does or look the way it does. You'll notice in the picture there are some differences between DNA and RNA in its structure. First of all, RNA is single-stranded. It does not have two sides to the ladder. It just has one side. Secondly, it uses a ribose sugar on the outside part of the ladder instead of deoxyribose. And last, it contains no thymine. Instead, it has a different chemical compound called uracil. So it follows the base pair rules the same way DNA does, except that adenine does not bind with thymine because it doesn't have it. RNA doesn't have any thymine. Adenine binds with uracil. So take a look at this structure. What would be the correct pairing to go with this DNA if we had an RNA attaching itself in order to take this code out to the ribosomes? If we follow the same base pair rule, adenine, but here's the exception, adenine binds with uracil, so this would be A, this one would start with U. T still binds with A. So if you have a T in the DNA, you still have adenine or A in the RNA. So we have U, A, U. G and C doesn't change. G binds with C binds with G, and G binds with C again. This is what it looks like when RNA is starting to take that message out to the ribosomes. The blue part, that's DNA. The DNA has split open. There's an enzyme that helps with that process. And then you have each of the bases that kind of get connected to the DNA wherever it's split apart. So we have A binds with T, C with G, G with C, U with A, and so on and so forth. As all the connections are made, everything gets linked together, and then the RNA travels out to the ribosomes, and the ribosome reads the code. As always, if you have any questions, I know that this is complicated, so please don't hesitate to send me a message, and hopefully you understood what we were talking about.